Hello students, welcome to our next lecture. Today we are going to start primary structure of dicotyledonous stem. In our previous lecture we have seen the primary structure of dicotyledonous root. So here we start the internal or when we take a transverse section of the stem of sunflower stem or any dicotyledonous stem, we will see the from periphery, that is from periphery up to the center, we will see the various types of tissues present in it. So from the epidermis and up to the pith. So there are three regions in dicotyledonous stem, that is epidermis, cortex and stele. So epidermal part is the outermost covering part of the plant body. Then under the epidermis up to the endodermis, it is a cortex part. That is, in cortex there is a presence of various types of permanent tissues, simple permanent tissues. Okay, so these transverse sections, it shows the outermost covering epidermal cells, then the middle portion, that is the cortex, which is made up of various types of permanent tissues, simple permanent tissues, and the center part, which is located at the center of the plant body. It is made up of stele, and stele is nothing but the combination of pericycle and vascular bundles. So vascular bundles, they are made up of complex permanent tissues, that is xylem and phloem, and at the center there is a presence of pith. Okay, so it is a continuation of the ground tissue. Pith is the continuation of the ground tissue. So stem, any stem of the dicotyledonous plant is made up of these three parts, that is epidermis, cortex and the stele. So let's look one by one. Epidermis, as in the case of root, we call it the epiblema, because in root the epidermis is known as the epiblema, or it is also known as the piliferous layer. In case of stem, it is the epidermis is the outermost covering or the outermost protective covering of the plant body or the stem. It also shows the unicellular or multicellular. It is in figure you can see the multicellular hair-like projections comes out from the epidermal layer or epidermal cells, and that multicellular epidermal structure is known as the hair of that epidermis. Or you can see, or you have seen that the sunflower stem shows a hair-like projections. So these hair-like projections is nothing but the prolongation or the appendages of the epidermis. It is a multicellular. In case of root, the hairs, they are unicellular. Okay, so in case of stem, it is multicellular. So multi-cells, more cells together to form a thin hair-like structure, that is known as stem hair. It is these epidermis, as you can see in the picture, it is compactly arranged and it is a single layered structure. These compactly arranged rectangular cells, they are present in a single layer, and it does not contain any type of chloroplast. So these cells, they are compactly arranged. Why they are compactly arranged? Because if there is a gap in between these two cells, that means if the intercellular spaces are present, then the transpiration or the that is the removal of water, you can say the evaporation of water takes place through this pore, and it may get harm to the plant, or it leads to the desiccation of that plant. So the water scarcity is there. So to avoid the transpiration rate, these epidermal cells, they are compactly arranged. And again, to give the protection, or to give the additional protection to the stem, these epidermal cells, they are covered by a thick cuticle, or it is the layer which is made up of waxy cutin. So due to the presence of this cuticle, these epidermal cells, they are more protective in nature, and also it avoids the transpiration, or the rate of transpiration becomes very low. Okay, so it is cutinized. This epidermal layer is cutinized. Okay, so as in case of root, it does not have any cuticle layer on it, because the function of root is to absorb the water. So to facilitate the absorption of water, there will not be any barrier in between these. So if the cuticle is present in root, so water absorption does not takes place. So but in case of stem, to avoid the water transpiration or to avoid the water evaporation, the cuticle must be present. Okay, so it is cutinized. Then multicellular hairs and stomata, they are also found in the epidermis. So stomata is meant for, it is a media for the gaseous exchange, and it is media for the transpiration. So gaseous to facilitate the photosynthesis process, plants needs to that is require the gaseous exchange process. So stomata is nothing but a pore. It is a small pore that is present through which the gaseous exchange and the evaporation of water takes place. So in young stem, there is a presence of stomata also. And it these epidermis cells, that is cutinized epidermal cells, it protects the inside tissue, it protects the innermost, that is cortex tissue, and all the underlying tissues, they are protected by this epidermis. So it is a outermost or protective covering which makes a barrier, or makes a physical barrier in between the underlying tissues and the external environment, so that the underlying tissues, they must be protected. Okay, so this is the epidermis. Then the next part, just placed below the epidermis, is the cortex. Okay, this cortex, now in case of stem, the cortex, it may be heterogeneous, but in case of root, we have seen that the cortex is homogeneous. So what does it mean? That cortex, which is made up of only single type of that is tissues, is known as the homogeneous cortex. But in case of heterogeneous cortex, there is a presence of more than one type of tissues. That cortex is made up of simple permanent tissues, and simple permanent tissues, they are of three types: parenchymatous, cholenchymatous, and sclerenchymatous. So in case of stem, there may be a presence of more than one type of tissues in the cortex, hence the cortex is heterogeneous. Okay, so this cortex region, it is lying just below the epidermis, and it is above the pericycle. So from the that is from below the epidermis up to the endodermis, the region is known as the cortex region. Okay, so just below the epidermis, there is a presence of cholenchymatous tissues. So the portion which is made up of cholenchymatous tissues is known as hypodermis. Okay, so it is present just below the epidermis. It is thick multicellular layer, that is the cholenchymatous tissues. As we have seen what type of cholenchymatous tissue is. Cholenchymatous tissue, they are somewhat thick walled, that is the thickening is at the corner of the cells. Okay, that is uneven thickening. Parenchymatous tissues, they are thin walled, but the cholenchymatous tissues, they are thick at the corner. So they are somewhat thicker than that of the parenchymatous tissues. So they are thick and multicellular, that is the they are present in one more than two layers. So it is a multi-layer structure, and the cells, they are round or oval in shape. This as you can see in the figure, that the cells, they are somewhat elongated structures, or they are made up of oval type of structure, and their walls, they have become thick at the corner. So this is the cholenchymatous. In some cases, in some stems, these cholenchymatous tissues, they are there is a presence of chloroplast. or the chlorophyll pigments. Hence they are helpful in the photosynthesis process also. It is as their cell walls, they are thick, they gives the mechanical strength and rigidity to the stem. So it gives protection to the stem. So first protective tissue is the epidermis, which is cutinized. Underlying the epidermis, there is a presence of Sclerenchymates hypodermis, which also gives the protection and the mechanical support to the plant. Okay, this is the 2 to 3 layered hypodermis is present just below the epidermis. Now next to the Sclerenchymates tissue, or next to the hypodermis, there is a presence of general cortex. So these general cortex, which is made up of parenchymatous tissues. Okay, so it is just lies below the hypodermis, below the Sclerenchymatous layer. You can see in the figure that just below the Sclerenchymatous layers there is a thin walled structure is present. Thin walled tissues or cells, they are present. So these thin walled tissues, which shows the intercellular spaces also, they are known as the parenchymatous tissues. So this general cortex, or you can say the ground tissue, which is made up of thin walled parenchymatous cells. So it is a multi-layered structure also, and made up of this all the ground tissue, which is made up of parenchymatous cells, and it shows also the intercellular spaces also. And the function of this ground tissue, or the function of this general cortex, is the to store the food material. Okay, as you know, the function of any parenchymatous tissue is to store the food material. Here is the same, that is in the stem of sunflower, the parenchymatous tissues also perform the function as the food storage. Okay, so this is the that is second part of the cortex, as you also can say is the general cortex. So below the hypodermis, this cortex, it lies below the hypodermis. So the innermost or the last part of the cortex is the endodermis. So this endodermis, as you can see in the figure, that the is the demarcation line in between the cortex and the that is the underlying vascular bundles. So this is somewhat barrel shaped cells, you can see in the figure, and it is labeled, that is you can see the endodermal region just below the cortex region as a single layered structure, which is the innermost layer of the cortex, and the cells of this that is endodermis, it is a barrel shaped and which are compactly arranged. And have you better know how the endodermis tissues, they are like, that is barrel shaped, they are compactly arranged, their radial walls, they are somewhat thicker than that of the outer and the inner walls. And these radial wall thickening, it is the known as the Casparian strips. It looks like a Casparian strips. So these Casparian strips or the radial wall thickening is absent where the protoxylem face, that is it is present when the cells of the endodermis, when they are present against the protoxylem, these cells, they are not thickened, they does not show any radial thickenings, and these cells, they are known as the passage cells. Why these cells, they are not thickened? Because the from that portion only the water movement takes place, and that portion of the endodermal cells which does not show any thickening, that cell is known as the passage cells, as they allow the passage, or they act as a passage and allow the movement of water from inner side to the outer side, or vice versa. These cells, they are known as the passage cells. They only those cells which are present against the protoxylem, that cells, they does not show any thickening. Okay, so all the endodermal cells other than the passage cells, they shows the radial thickenings, or known as or it shows the Casparian strips. Okay, so this is from up to the endodermis, the below the epidermis and up to the endodermis, the portion which shows in the figure, it is known as the cortex region. So cortex is divided into two parts, hypodermis and the general cortex. When it is a heterogeneous cortex, then it is differentiated into two parts, or it may be into three parts also. So the upper portion that is just below the epidermis, it is known as the hypodermal part. Hypodermis, that is made up of sclerenchymatous tissues, and the other part that is known as the general cortex, which is made up of parenchymatous tissues. So two tissues together form a cortex region, and the innermost part of the cortex is known as the endodermis, or it is made up of endodermis. Okay, so this is up to the cortex. Epidermis is the first part, then second part of any that is internal structure made up of cortex region. Now the there will be start of steel portion. So these three regions are present in any internal structure of plant. That is, first is epidermis, then the second part next to the epidermis is cortex, and the third part that is present in the center of the plant body is known as the steel region. So let's look at the steel. Okay, now as you've seen in the figure, that is the demarcation line is endodermis, and below the endodermis there is the portion up to the pith is known as the steel part. The steel, what is included in the steel? Which type of structure present in the steel? Yes, the steel contains pericycle, vascular bundle, and the pith region. So these three parts together form a steel in any plant. Okay, this steel, which is differentiated into these three parts, they are known as the pericycle, vascular bundle, and the pith region. So first part, that is just below the endodermis, as you can see in the figure, that is the sclerenchymatous patches are there. These patches, that the dark patches, two dark patches you have seen in the you can see in the figure, and which the cells of these patches, they become very thick. Okay, so they are evenly thickened, and that tissues, they are known as the sclerenchymatous tissues, and they are the dead tissues. Why they are dead tissues? Because their walls, the deposition on their walls is the lignin material, and due to the lignin material, they are impermeable to water movement, and as they does not get water, or there is no availability of water, hence the protoplasm, they become die in the these sclerenchymatous tissues, hence they are dead tissues. Okay, so these sclerenchymatous patches you are seeing in the that is picture, it is known as the pericycle. So this pericycle, which lies just below the endodermis, is composed of sclerenchymatous and the middle portion, which is made up of parenchymatous. So there is not a continuous layer which is formed by the sclerenchymatous. This pericycle is not continuous. So there are some patches of sclerenchymatous tissues, and in between these patches there is a presence of ground tissue, which is made up of parenchyma. So two types of tissues, they are present in the pericycle, sclerenchyma as well as parenchyma. So it is composed of this pericycle, which is composed of sclerenchymatous patches and the parenchyma. Okay, so this is the multi-layered structure. You can see in the picture or the figure, it is a multi-layered structure. So pericycle is made up of more than one layer or more than two layer. It is a 2, 3 or multi-layered structure. Okay, so this is the pericycle, which is present just below the endodermis. Now uh, as we go further inside to the stem body, we see that is just below the pericycle there is a presence of vascular bundles, a patches of vascular bundles. These vascular bundles, they are somewhat different from that of the root, as we have seen. Okay, so these how which types of vascular bundles they are present in the stem? Yes, these are conjoint, collateral, enlarged and open type of vascular bundles. What does it mean? conjoined collateral. That means these vascular bundles, it is made up of complex permanent tissues, that is xylem and phloem. Okay, in xylem and phloem there is a presence of cambium also in case of sting. So these vascular bundles, when there is a presence of xylem and phloem on the same radius or the same radii, that is the xylem is present, that is inside there is a xylem presence, and upper to the xylem there is a presence of phloem. That means they are present on the same radii, on the same line, that is upper xylem and inner phloem. But in case of root, we have seen that this xylem, they are present on a separate radius, and the phloem, they are present on the separate radius. That means they are present on the different radii. But in case of stem, they are present, that is xylem and phloem, they both the complex tissue, they are present on the same radii. Okay, so this is the difference. Hence they are known as conjoined collateral. Okay, these are the conjoint and collateral. Then as we have seen in the root, that is there is a exarch condition. So when the protoxylem of the vessels of the xylem has two types of vessels, that is the large mouth part, that is the metaxylem, and the smaller one is known as the protoxylem. That is the enlarge and exarch condition, it depends on the position of the protoxylem tissue, that is the protoxylem cells. When the protoxylem, they are present towards the direction of periphery or towards the direction of epidermis, then it is a exarch condition. But when the protoxylem, they are facing towards the central part of the stem or towards the pith part of the stem, then it is a enlarged condition. Enlarged means in, and exarch means towards the outside, and enlarged means towards the inside. Hence in this stem, internally you can see in the picture that these small vessels, that is the protoxylem, they are present towards the pith region, towards the central part of the stem region. Hence the that is the it is the condition is known as the enlarged condition. Okay, and open means what? Open means the when the cambium or the meristematic tissues, or you can say the that is the continuously dividing tissues, when they are present in the in between the xylem and the phloem. You can see in the picture, that is a multi-layered patches are present, that is the label it is labeled, that is cambium tissue. That is this cambium, when present in between the phloem and the xylem, then it is the known as the open vascular bundle. Open means what? Open to divide. That means these dividing tissues, continuously dividing tissues, when they are present in between the xylem and the phloem, then that part, that or that organ of the plant is open to divide. Hence it is known as the open. That is the division is present in that tissues. But in case of root, there is that is the cambium is absent. Why? Because further development or the further division of the cells, or you can say the secondary growth is absent in that root. Hence in those parts where the secondary growth takes place, only those parts these multicellular that is the you can say the continuously dividing cells, they are present, that is the meristematic tissues, they are present in that part. So in stem there is a presence of secondary growth, hence the cambium tissues, they are present in between the xylem and the phloem. So this is the vascular cambium. Okay, when the cambium or this meristematic tissues, when they are present in the that is a phloem and the xylem, okay, this is known as the intrafascicular cambium. Intra means in between these xylem and phloem. Okay, So this is the in between, when in between the xylem and phloem these meristematic tissues are present, they are known as intrafascicular cambium. Okay, so these vascular bundles, these conjoint, collateral, enlarged and open type of vascular bundles, they are present in a ring like structure. They are arranged in a ring. Okay, a complete circular ring is present, that is, but in case of root they are scattered everywhere. They are not arranged or in a rings like structure. So but in case of stem they are arranged in a ring structure. Okay, so vascular bundles, that patches of vascular bundles, they are present in a ring like manner. Okay, and these vascular bundles contains phloem. The outer part is known as the phloem, and the inner part is known as the xylem, and in between this phloem and xylem there is a presence of meristematic tissues layer, multicellular layer, that is known as the cambium. Okay, so these vascular bundles, they are associated with the pericycle patches. As you can see, these pericycle patches, inside to the just inside to the that patch there is a presence of structure that is known as phloem, and inner to the phloem there is a presence of cambium, and the inside the cambium there is a presence of xylem, that is metaxylem and the protoxylem. Okay, so this in between these two parts, in between these two vascular bundles and in between these two pericycle patches, there is a presence of continuous, that is continuation of the parenchymatous tissues. Okay, so as you can see in the picture, there is a continuation of parenchymatous tissues. They are known as the medullary rays. Okay, they are present in between the parenchymatous patches and in between the vascular bundles also. Okay, as you can differentiate these tissues from this pericycle and the vascular bundles. So in between these two pericycle patches, that is sclerenchymatous patches, thin walled parenchymatous cells are present, or you can see the continuation, you can see the in between this phloem also parenchymatous tissues are present. In between this cambium there is also a presence of parenchymatous cells, and in between these xylem tissues also there is a presence of parenchymatous tissues, and these parenchymatous patches, they are continue with the pith, up to the pith. From pericycle it is continue up to the pith. Okay, so each vascular bundles which is composed of phloem, then cambium and the xylem. It is the conjoint, collateral, enlarged and open type of vascular bundles. Okay, clear? The next part, that is the uh, vascular bundles, they are present in the arranged in a ring. These vascular bundles which is composed of phloem, cambium and xylem. So let's look at the phloem tissue. It lies towards the periphery and consists of, periphery means it lies just inside the pericycle. Okay, and it consists of again the complex tissue which is made up of four types of different types of cells. That means phloem is made up of sieve tubes, phloem is made up of companion cells, phloem parenchyma and sclerenchyma. That means phloem fibers. Okay, so it consists of four different types of cells. So this phloem is also consist of four types of cells, that is sieve tubes, companion cells, phloem parenchyma, and there may be a absence of phloem fibers in many dicotyledonous stem. Okay, but these three cells, that is sieve tubes, companion cells and phloem parenchyma, they are present in all the types of stems. Okay, then in sieve tubes there is at the end of the sieve tubes there is a presence of sieve plates. Okay, which is they are perforated cells. Okay, so phloem, that is the function of phloem is to conduct food from uh, the region where they manufacture, or from the region where they produced, to the other parts of the plant body. Okay, So this sieve tubes, that is through these sieve tubes the food is conducted or transported to every part of the plant body. So it acts as a pipeline of the plant body. Okay, so then the cambium part is the below the phloem tissue. There is a presence of, in between the xylem and phloem, the cambium or the meristematic tissues are present, and this cambium is known as the intrafascicular cambium. It is single to many layered. Okay, so in this picture you can see the many layered or the multi-layered cambium. Okay, then next to the xylem, it consists of, xylem is again made up of four different types of cells, that is xylem vessels, tracheids, xylem parenchyma and xylem sclerenchyma. So the together these different types of cells, the xylem tissue is made. Okay, so xylem has two types of vessels, metaxylem and the protoxylem. Okay, these are thickened dead tissues, and they forms the wood structure of the plant body, and they give the protection to the plant body as their cell walls they are thickened. Okay, so this is about the vascular bundle. So as I said earlier, that in between these vascular bundles and in between these pericycle patches, there is a presence of ground tissue, a continuation of the parenchymatous tissue. So this ground tissue in between the two adjacent vascular bundles, a wide strip is present, that is polygonal parenchymatous tissues they are present here, which are extending from the pericycle up to the pith. That portion is known as the medullary rays, or you can say the pith rays, because it is a continuation of the pith rays up to the pericycle. Okay, so this is the medullary rays. They are present they are present in between every vascular bundle or every pericycle. So these medullary rays you can see in the whole ring structure of the that is vascular bundles, the steel. This steel or the vascular bundles, they are present in a ring structure. So the in between the every vascular bundles there is a presence of medullary rays. Okay, so this is the medullary rays or the pith rays, which is a continuous extension of pith up to the pericycle. And what is the function of these medullary rays? As they are made up of parenchymatous tissue, the function of parenchymatous tissue is, yes, the food storage of food material. Yes, these medullary rays, they also act as a food storage cells or the tissues. Okay, so the last part or the innermost part of the steel, that is the pith part, and it is you know that it is made up of parenchymatous, thin walled parenchymatous tissues. This thin walled parenchymatous tissue shows the gap in between these tissues, that is the intercellular spaces are present. They are large in size and stores the food material. In the older as the stem get older, the pith is absent, or the pith is not present at the older part of the stem body. Okay, there is a presence of gap is present towards the center region. Okay, so this is up to the, that is the anatomical or the transverse section of the sunflower stem from the epidermis up to the pith. How the tissues located, how the different types of tissues they are located internally from periphery up to the pith, you can now better understand. Okay, so three regions, we can differentiate this internal structure into three regions. The first part is the epidermis, then the second part is cortex, and the innermost part is the stale region. Okay, so every anatomical structure is differentiated into these three regions. Okay, when you write any anatomical structure of the plant body, you only you must be divided these portions into three regions: epidermis, cortex and the stale region. Then you can elaborate that point. Okay, so this is the about the structure of sunflower stem. or you can say the primary structure of dicotyledonous stem. So what is the difference between root and the stem? Yes, root, the epidermal portion, that is the outermost covering, is known as epiblema or piliferous layer. In case of, it is known as the epidermis. In case of root, the hairs which arises from the epidermal portion, they are unicellular in structure, but in case of stem, they are multicellular in structure. Then come to the cortex region. The cortex of the root is homogeneous, but in case of stem, it is heterogeneous. Heterogeneous, that is made up of more than one type of tissues. So here stem is made up of collenchymatous as well as parenchymatous cells. Okay. Then come to the next part, that is the stele part. Okay. The stele, in case of root, the pericycle is not made up of, that is there was not any patches of the pericycle. It is not multi-layered, it is a single layer. It was a single layer. But in case of stem, the sclerenchymatous pericycle is present, which is present in multi-layered structure, and it shows in it present in patches. Okay. So the vascular bundle, in case of root, it is radial vascular bundle, but in stem, it is conjoined, collateral, then endarch and open. In case of root, it is exarch. In case of stem, it is endarch. Then in case of root, they are closed type vascular bundles, but in case of stem, they are open type. So open and closed. What does it mean? That when the cambium tissues or the you can say meristematic tissues, when they are present in vascular bundles, that vascular bundle is known as the open type, and when they are absent, they are known as the closed type. Okay. So this is the difference between root and the stem. And in case of root also, this arrangement of vascular bundles is scattered, that they are not arranged in a specific manner. They are scattered everywhere in the stele portion. But in case of stem, they are arranged in a specific manner. They are arranged in a ring like structure. So this is the differentiation, or when you see both the slides, both the transverse section of root and stem, you can easily identify now the which slide is of root and which slide is of stem. So internally, as you differentiate externally, also internally you can differentiate the root and the stem structure. Okay. So this is about the today's lecture, that is the primary structure of dicotyledonous stem. Further, or in our next lecture, we will see the leaf of dicotyledonous structure. Okay. So up till now, stay tuned with us, and we will meet in our next lecture. Thank you very much.